All right, so I want to say thanks for everybody coming out on the night before Thanksgiving. I know a lot of people have other obligations, family stuff, so really appreciate you all coming out here tonight. We hold a STEAM event every month at TSBQs, also at Cafe Pace out in Monroe, in Bellevue. And then just yesterday we got a new location in Tacoma at B-Sharp. So we're starting to kind of grow the STEAM event community, so that's really great. And we'd love to have your regular participation, so thank you very much. One's only been doing this for about almost 5 months now. So it's growing slowly but consistently, so we're pretty happy about that. How the evening goes is we have a 20-minute, half-hour presentation by an expert, and then it's really about a question-and-answer process. It's a discussion. It's a conversation. This should never be a lecture. If you want a lecture, go to the classroom. This is a conversation and discussion. So we really appreciate our speakers coming in and being open, candid with everybody. So without further ado, Hannah Chapin is a PhD with the Department of Biochemistry at the University of Washington. She came to UW through Yale University, where she got a PhD in cell biology. I don't know the first thing about biology, so I've got a lot to learn about this. I'm a space nerd. In the laboratory, she has extensive experience in designing protein cleavage-based assays, and when away from the bench, she has passion for and experience in teaching students, the public, and her peers about the wonders of the natural world. She came to us through Hive Bio, so those folks have been really great to kind of opening some doors for us. And let's talk about
yeah, worms and getting old. Inevitable decline. So thanks for the introduction, and thanks to you and Gwen for putting this together. This is a really awesome event to have happen. I think it's a good thing to grow in the city. So you mentioned a little bit about my scientific background. I came here via the East Coast and the Northeast, and then came out to Seattle for my postdoc, which is sort of this scientific apprenticeship after you get your PhD. And I got into aging research in part because that's where the money is, and there was a funding opportunity, but that funding opportunity got me into a community of aging researchers at the University of Washington. There's actually a lot of people doing this stuff at UW. I took a summer course at Woods Hole out in Massachusetts in August, and so was immersed in aging thinking and research for about a month. And so that was part of the reason I decided to start sharing all of that stuff I learned with other people. So I want to tell, first I want you to know at the end of this night that you can study how we age, that it's not just a thing that happens. It gets you a little bit familiar with the kinds of ideas people have about why we age. I am going to show you some results, because that's not a thing some of you see daily, some of you see a lot of it, and some of you don't see very much of it. So I'll go through some results and get you a sense of how I study it on my scale, and finally open it up. Share what I know, field questions. We can sort of think collectively about what are the big questions in this field. What brings you guys here? Some of you I know. Does anybody have any particular interests or specific areas of fascination? It's a small enough group I won't put you on the spot, but as we go through the evening, feel free to speak up and ask questions as we go. All right. Why do you die when you die? If you die of natural causes is a really challenging question to answer. And there are a lot of reasons. And we really want there to be a single reason, right? We'd really like there to be one thing we can fix so that we live forever, and that's not the way it happens. A bigger question, in a way, is what determines how long any creature lives. And this gets to a lot of the comparative biology that goes on as we try to understand why different animals live different lengths of time. There are a lot of things that change as you get old. This is the long list of vocabulary that may or may not mean anything to you, but you will find an expert or 2 or 7 in the field who devoutly believe that any given one of these answers is the reason we age. So you could find anybody who will talk your ear off about any one of those topics. So there is no single answer, but there are a lot of hypotheses. And so the challenge then becomes trying to figure out which of those hypotheses are useful and to what extent each of them are useful. Lots of hypotheses. So one idea, and I've led off with this because it's kind of easy to grasp. In a way, this feels a little bit woo-woo, right? You run out of magical juice. But there is a theory that you have a defined number of heartbeats, right? Because elephants have a fairly slow heartbeat, and mice have a fairly fast heartbeat. And so when you look around the animal kingdom and you say mice die faster than elephants, maybe they run out of heartbeats faster. And that sounds sort of silly, but then you graph it and it turns out to be true. So you've got here life expectancy in years. So this is how long the species on average lives. Up here on the y-axis is heart rate, beats per minute, and this is a log scale. So it's actually been compressed a lot. So there's actually big differences in heartbeat. And you can see that as heartbeat goes down, life expectancy goes up. And we're a bit of an outlier out here. But on the whole, if your heart beats slower, you live longer. Which is kind of weird, but I'll talk about some reasons that might be true. It plays a bit into metabolism. So metabolism is how fast your body goes, essentially. It's how fast your cells are running, how quickly you're turning food into energy and energy into actions inside your cells. So metabolism as body mass, this is just like the other graph, except now this is energy that you use per day. So how much energy are you using? The less energy you use per day, the longer you live. So elephants use less energy than mice, who are scurrying around a lot and trying to stay warm and fed, and elephants sort of lumber around and live a really long time. This is also true if you look at body mass. So this is a graph somewhat like the other one I just showed, but this is body mass in years. So the bigger you are, the longer you live. So the bigger you are, the slower you live, and the longer you live. And this is true if you look within mammals here in blue. All the blue dots are mammals. All the red dots are birds. And so people use this information in a couple ways. First, they say, why does metabolism affect lifespan? Because that's something that apparently happens. The other is, what can we learn from outliers? So how can you find dots on this graph that are way off from where you'd expect them to be? What kind of species is that, and what can we learn from studying that species? So bats, it turns out, live longer than you expect. Bats are about the size of rats, but they live way longer. And so people are studying bats, trying to understand what it is about bats that makes them live longer than something of comparable size. So this sort of rate of living hypothesis goes back to oxidative damage, among other things, but we'll oversimplify it for tonight's purposes. Oxidative damage is damage that comes from producing energy. So your mitochondria, you may have heard the powerhouse of the cell, is what turns glucose, food you eat, into energy by way of pumping protons. There's a bunch of energetic transfers there that result in free radicals being made, oxygen radicals. You may have heard of antioxidants. You can buy them in pills, eat a lot of blueberries, that kind of thing. So you're right, pound them back. So those antioxidants are supposed to neutralize those radicals, because those radicals run around your cells causing damage. So the idea is, the longer you metabolize, the faster you metabolize, the more radicals you make. Radicals, right? Protests, and they got to pull out the barricades. The more free radicals you make, the more damage you get in your cells. The more damage there is in your cells, the more likely you are to fall apart, which is essentially age. So that's the oxidative damage theory in this whole list of ideas. So that's one example of one theory for one possible explanation of a piece of why we age. I now want to talk about two other pieces. This protein aggregation, we don't get boxes. All right, proteins aggregate and organelles become less efficient. So this is where my work comes in. I'm going to talk about my work for 5 minutes or so, 10 minutes, and then we'll open it up. Are there any questions, though, just about these theories and rates of living? And
Are the
Yeah.
humans an outlier on that chart? It showed the 70 to 80 years if you were to look at historical data.
Right. Are humans an outlier on the chart of age and body mass and rate of living? Ish, yeah, a little bit. But then the question is, you know, what measurement of human age are you going to use, right? Because you could, if you try to find the oldest living documented human from every point in time, it ends up being approximately a flat line, with I think a weird spike for Methuselah, who like lived 200 years or something. But for the most part, yeah, right. Some guy put it on a graph last week in a talk at work, so it must be true, right? Yeah. So you can't, we have a maximum lifespan-ish. Average lifespan, though, is the thing that the rest of us care about, right? So we are enough of an outlier that we consider ourselves special, but we're trying to figure out what it means.
There are some animals that don't experience senescence. And what are your thoughts about that?
So there are animals that don't experience senescence. So senescence is death, cellular death. You're talking about animals that don't seem to decay as much.
Lobsters?
Lobsters. I don't know much about lobsters and aging. You've got me on that one. So the idea is that you have to have cells die and then clear them out in order to make room for the new. And if you don't, you end up with a bunch of cells that accumulate damage. So we'll wake it up again when we need it. Somebody's probably studying lobsters. I know that people are for sure studying naked mole rats, which live for a crazy long time. So most rats live like, I don't know, 10 years, 5 years, something, 10 years. And naked mole rats, about the same size, live like 50 years. Somebody's trying to start up a colony of them, which is very challenging because you don't know how old the wild-caught ones are. You've got to keep them alive and happy for decades. And grad students don't want to work on aging in these animals because they'd never graduate. So the weird animals that do funky things are doing funky things for a reason. And part of people researching those model systems are trying to figure out, is that relevant for other systems?
You always thought myself working at NOAA for a while.
Working at?
NOAA?
Oh, nice. Yeah.
So I just happened to find that out.
I will have to look into lobster aging. I don't know much about that. That's a good question.
Is there a difference among, like, or between mammals and birds and reptiles and insects, or is it all across the board?
For the correlations showing, I showed for mammals and birds, and those hold true. Generally, it's hard. Insects, you can't get as much of a weight change, thank goodness. So it's hard to compare really, really big to really, really small in those categories. So I don't know if it holds true over the small scale, and I don't know how much we could interpret from that.
Like, I mean, I can't think of the scientific lobster right now. I mean, it's a totally different
Right,
animal.
yeah, it is. Crustaceans, yeah, and I don't know where crustaceans fall on that graph. That is a really good question. I'd say I'd get back to you, but it would be an empty promise. But I will look it up on my own. Give me your email. I'll see if I can find an answer for you.
Yeah, I have a question
Yes.
from the previous slide.
Yes.
You know, and you don't have to answer this now, but you know, they will sell you. They make billions of dollars off selling people antioxidants.
Yep.
Nobody's been shaking a can of organelle deficiency chips in front of me, or protein disaggregators, or telomerase lengtheners. So as you go along, I'm curious, like why those all not, you know, because everybody wants to live forever.
Because biology is really damn complicated.
Right, totally. But marketing,
They would sell you telomerase, but they can't figure out how to get it. I'm sure you could buy it, actually. I bet if you go on the internet right now, you could buy telomerase. It'll probably give you cancer, right, of some flavor. Most of these will probably give you some sort of cancer. I mean, so protein aggregation, that's Huntington's or Alzheimer's. So people are trying really hard to make things that will break apart those aggregates. So it's not for lack of trying. Not for lack of trying. And the DNA accumulates mutations, so that's sunscreen. They do sell you DNA damage preventers. And that will help you live longer. It's true. But yeah, people are trying to market this stuff, and they will.
oh yeah,
as soon as they can. Yeah. So I'm going to explain some of my work. The big theme in this is that aging is essentially a descent into chaos. If you want to go to the physics level of it, entropy is increasing and all of our cells are slowly falling apart, essentially. So these are the pieces of a eukaryotic cell. And some of these may be familiar and some of them may not. There's a nucleus where the DNA lives. There are these mitochondria, those energy powerhouses I was talking about earlier. There's some other little vesicles and lysosomes and microtubules. The take-home message from this slide is that every process in a cell has its own compartment. So every process has a spot it happens. These restrictions require that there be some order inside this chaos that is a cell. So it has to be divided up into order, and maintaining order takes energy. Just like your house sort of falls apart if you don't pay attention to it. Your cells would fall apart too if they didn't put energy into making sure proteins stayed where they're supposed to be. And the cell puts most of its energy towards making sure stuff is where it's supposed to be. Order decreases over time, and that's the take-home, really, for a lot of aging, is that over time, the careful order that the cell has started with or built up evaporates and stuff goes bad. So part of my work has been trying to figure out how cells keep functioning by getting rid of the stuff that no longer works. So if you can degrade proteins that aren't working, organelles that have stopped functioning, aggregations, if you can keep cycling through the contents, it means you can break apart those pieces that are broken and put them back together into new versions that are functional. This process is called autophagy. So this is my statement. I'm going to go through this now piece by piece. This is what I've spent the past 4 years working on. Yeah, it boils down to this sentence. So the flux of autophagy varies by tissue and by type of stress in C. elegans. All right, we can go home. That's it. I want to convince you, though, that this is actually interesting and that this is powerful for some valid reasons. So I'm going to explain it phrase by phrase. First, I want to explain what the flux of autophagy is. It sounds like something you should bottle and sell. Would you like some autophagic flux? So autophagy is self-eating, autophagy. It actually means the cell takes pieces of itself and breaks them down. So this happens in the cytoplasm up here. It's forming this double membrane bound vesicle. It's like a bubble. This is kind of like seine net fishing, but without the helicopters and without the dead dolphins. It's sort of indiscriminately taking a bite out of the cytoplasm and degrading everything that's in there. So the pieces of the cell, the cytoplasm, proteins, organelles, get surrounded and then delivered to the lysosome. And the lysosome is the garbage disposal, is the garbage can. It's where all the enzymes live that break stuff down. The thing in Star Wars. It will, yeah, it's sort of the cellular black hole, except stuff comes out and can be used again. So autophagy, the self-eating process, takes chunks of cytoplasm and breaks it down. All right, so this clears protein aggregates and dysfunctional organelles. So in theory, you could keep everything nice and healthy and you'd live longer. We needed a new assay because this is how people have been studying it. And I don't even need to explain very much here to tell you that these pictures show an increase in autophagy. I did not want to spend my days doing either of these assays. So this is looking at EM sections. You can actually slice up a worm and look for structures, physical structures that indicate autophagy is going on. And in this case, we're looking for green dots in green cells. No kidding. This is the state-of-the-art assay. So these are not tissue specific. They're hard to do. They're impractical. And they measure static structures. So autophagy is this dynamic flux, and these things measure static structures. So I decided I wanted to measure the process, the movement through the process, and I didn't want to have to count dots. So I'm going to go into technical jargon for a minute. You can ignore it until I get to the picture when I show you what to look for. The idea here is that we've got two fluorescent proteins. Think of these like the GFP. Have you heard of GFP with jellyfish? So they're like 2 GFP molecules. These are really tightly bound, compact little molecules. So I link 2 of those up with a really floppy linker. So tightly folded protein, floppy linker, tightly folded protein, and then it's hitched to this LGG1, which is an autophagy protein. So it's involved in forming that membrane. So it gets taken along for the ride, and in the lysosome, where it's exposed to all those enzymes that are going to chop stuff up, the floppy linkers fall apart, those tightly folded proteins stay bound together, and we end up separating them into singles. So what I can do is run this all on a gel. I separate it by size. I then get a gel with big proteins at the top and little proteins at the bottom. I then, using some antibody tricks, look for the GFP. So I'm looking for that tightly folded protein. And I see it here at the bottom and at the top. So up here is the doubly tagged version. Down here is the singles. And this band right here at the bottom means autophagy happens. Okay. So I went through that whole thing to show you that I'm going to show you a lower band and say, look, there's my signal. So my signal is the result of proteins getting all the way through to the final step. All right, varies by tissue. Tissues, worms and humans. Same, sort of. Mostly, I mean, the key parts here are that worms, kind of like us, are a reproductive tract supported by a digestive tract. Nematodes, though, are really easy to work with. There's a large literature about aging, and there's not very much known about autophagy. So I can use promoters. So I use a trick of DNA, genetics. I have a promoter that will express a protein in just the neurons, or just the pharynx, or just the intestines. So then, when I look at that whole plate of worms, I'm seeing a signal from what's going on just in intestinal cells. So I've got tissue-specific expression here. You can see that the neurons, the pattern of the protein in the neurons looks different than the pattern of the protein in the intestines. So it gives me pretty pictures. And then I can run them out on a gel. So I take a whole tray of worms, squish them up. I treat them if I want to. I run them out on a gel, and I get this. We've got full-length protein up here and broken up monomers down here. And even not telling you which tissue is which, you can see that it's different. So different rates of autophagy are happening in different tissues. If I label this for you, we find out that the intestine has a lot of autophagy and the hypodermis has a lot of autophagy. I don't know why. And other tissues have fairly low rates. Hypodermis is skin. Yeah, skins. So we've got ubiquitous, so everywhere, intestine, hypodermis, which is skin, pharynx, which is like the throat, kind of this big elongated throat, muscles, which are the body wall muscles, and then neurons, which are like your brain. Varies by tissue and type of stress. So we'll get to the type of stress. Does anybody have any questions first? So I've been blathering a long time.
I have a question.
Yes. Chemical bonds.
As worms age,
Yeah.
can you do this in different age worms?
Yeah.
Does it show up in different places?
Ooh. The amount of autophagy happening in tissues changes with age. Yes.
But does it ever disproportionately go to neurons, like in a really old worm? If the worm had.
Yeah. So you and the reviewers would both like more information about that. So it's it's a good question. The question is, does it happen in a new tissue as they get old? Well, so I see it in neurons in old worms, and I don't see it in neurons in young worms. So it appears, for other reasons, I think that it's going on in neurons even in young worms, and I just don't see it because the degradation is happening too fast. The lysosome actually chews through my reporter before I can see it. So it's almost like it's too effective in neurons, which is kind of cool because neurons need to be functioning very specifically.
Like neural plasticity or whatever kind of slows down.
Yeah.
As you get older.
Right, everything doesn't work as well. Right, so maybe it's not refreshing the fleet as much, right? Like maybe it's accumulating stuff. Yeah. Yeah, I don't know. Can you stop autophagy? Yeah, so you can You can turn it off by using RNAi, so you can knock down a couple of the key proteins. And I've done that and shown that my reporter goes away. You can also add some drugs that deacidify the vacuole, that final end step, and I see it accumulate. And that's how I checked the speed, because I deacidified the vacuoles. I prevented the degradation and saw that it built up. Yeah. All right, I'm going to kind of blaze through this. It'll wake up. There you go. I'm just going to give you the punchlines. We can go through it if you want, but I want to talk and hear more and talk less from you guys. So I tried 3 different kinds of stress. Anoxia, so no oxygen, starvation, and age. So you kick them all or die. I mean, we do a lot to these poor worms. The bottom line is that in anoxia, autophagy goes up just in the intestines. Nice graph here if you want to look at it. In starvation, it goes up in all tissues. Basically, you can see a faint band there where you couldn't see faint bands before. And in age, it goes up in most tissues, and it has this really funky switch that happens in early adulthood. So there's something about the transition from juvenile to adult that induces an increase or a change in autophagic rate. There's this big literature about how proteostasis, so the ability to keep proteins functioning and happy when you get old, changes. You're worse at it. So I don't know. I presume this is playing into that somehow. And the question now is how. So I'm working on that. Give me another 4 years. So the summary of all of this is that I've got a new assay. It's technically accessible. Anybody can do it. It's tissue specific, and it's not restricted by age. I've shown that tissues have different baseline levels of autophagy, and that this is affected by stress and tissue. And why do we care? So there's a bunch of cases in which it'd be really nice to know how different tissues respond to different stresses. Ischemia is anoxic shock, basically depriving oxygen to a piece of your heart or a piece of your kidney. It's due to blockage in a blood vessel or traumatic injury. If we already know that your kidney has a different amount of autophagy going on than your brain, what can that tell us about how we treat ischemia in each of those organs? Aging, obviously, that'd be interesting. If tissues age at different rates, would it make more sense to target treatments or therapeutics to one tissue over another? Protein aggregation, so Huntington's, Alzheimer's, things that involve aggregation, people would love to be able to upregulate autophagy to fix those. And then, of course, there's the cancer connection, which you always pull in every scientific talk pretty much, but it's true that autophagy goes up in some cancers and down in other cancers, and why does that happen and what effect does it have? We don't know. So I'll put some of these questions up there just to kind of get your brains thinking. There's a lot we still don't understand about how aging works. So do tissues age differently? Why can I have exactly the same mutation as somebody else and I get cancer and that person doesn't? How do we fund aging research if it's not a disease? Because we have a very disease-focused funding structure, and we don't want to pathologize normal processes, neither do we want to deprive them of research attention. The difference between lifespan and health span. I want to extend lifespan, or excuse me, I want to extend health span. I do not want to extend lifespan without increasing the healthy years. So if you're going to be healthy until 70, I personally would rather die at 70 than live to 100 just for the sake of increasing lifespan. I'd like to bump up health span. And finally, there's a lot of questions about models, right? Monkeys versus mice versus rodents versus pick your favorite worms. So a quick thanks to Gwen and the steam vent folks, but I want to now see what questions you have. Yes.
Do worms have an immune system?
Do worms have an immune system? They do.
And can you look at the immune system as a tissue and as it relates to autophagy? So
the idea is how does the immune system respond, and what does the worm immune system look like? It's very rudimentary. Okay. So it's not as highly developed as ours. It's mostly innate. And a lot of it has to do, and I think this goes back to autophagy in the intestine, of degrading intruders, right? Because the intestine is the interface between the bacteria they eat, some of which want to colonize the worms. If the bacteria could get into the worm cells, that's a kind of happy place to live. So part of the reason I think autophagy is going on in the intestine is as a sort of immune, well, defense anyway, mechanism. Yeah, but I don't know how we can measure it in those. I don't know whether I don't think there are cells specific for immune response. So that'd be an interesting question.
Okay, I'm going to be passing the mic around for questions, but I always start with, imagine, how long have you been at this?
A decade.
Okay.
Well, it's science.
So imagine 10 years out, then 20 years out, then 30 years out. What does the future look like in this field? And how does that impact us? Like, you pull out your crystal ball, let's see what you got.
Right. I'll say it's some telomerase. So it's going both bigger and smaller scale. So science, I think this is safe to say in multiple fields, and I'd be interested to hear what other people think about their fields. Many of the studies now are looking at hundreds of, potentially thousands of people and saying, what can we learn from the population level about the effects of specific mutations or specific types of reactions, right? It's hard if you find one person, that's a curiosity, but if you can find 10 people, that becomes almost a significant data point. And if you can understand how my particular differences that separate me from you affect my aging versus your aging, it allows you to personalize medicine, right? So that would be the sort of pie in the sky, money producing way to interpret this, is that it would be personalizing in that sense, using genomic information or large scale population information. The other thing we're doing is going small. So that particular cell, but what about stem cells in your, muscles versus stem cells in your heart? And how do we target therapeutics or interventions to one subpopulation of a subtype of cell? How do you get super specific in targeting interventions? Because it's becoming clear that the sledgehammer is a really good way to mess things up and doesn't fix very much. And so fine-tuning, I think, is where we're going. And maybe that's the real answer. We're fine-tuning, but there are two different approaches to get there.
Okay. You've been studying this for a while. I presume that you're going to fight. I just turned 47 3 weeks ago. So I do what I can to, not slow the clock, even if I can't turn it back. Do you do anything in your own life to extend your life? Is there anything that you do after the research that you've done, the experiments that you've done, the community that you are involved with, the researchers? Is there anything that you do to extend your own life?
No.
Okay.
I mean, you can find people who do CR, caloric restriction. So I do know researchers at UW who skip lunch every day because intermittent fasting, or they don't eat on Tuesdays or something. Intermittent fasting is one of the few interventions shown to prolong life. Exercise is also one of the few interventions shown to have much of a reproducible effect. So I'd like to work.
Okay.
I mean, I stay active. If knowledge was enough to have us change our patterns, we'd all be living perfect lives, right? So there's way more to it than that. But mostly it's the things you hear, live right and get sleep and exercise. So I'm doing those things. I'm trying.
What
Yeah.
about the theory about insulin, modulation? People who fast have seemed to have, big spikes in there, and that seems to cause other issues.
Yeah. So it's the downside of fasting, the insulin. If I understand you correctly, the question that insulin is seen as a bad thing to have out of somewhat equilibrium, and fasting
The large fluctuations.
cause biggest fluctuations.
Say that,
Yeah.
Kept at a moderate level rather than having large spikes.
Right. Yep. And I think this gets back to you will find 10 people who devoutly believe that it's a problem and 10 people who are convinced it's the solution. So yes, there is something weird there about fasting and spikes and the dramatic changes that has on physiology. And for some people, it works really well. The more studies they do, the more they find out that caloric restriction helps 60% of the people and doesn't help 40%. and you don't know which percent you're in until you know, and you may never find out.
You don't want to give me that.
Yeah. So I don't know. And that's one of the things people are questioning, right? Is there a way you could even out those fluctuations? Just got the microphone. What would happen if you evened out those fluctuations? Would it be more or less effective? And I don't know. Yeah.
Okay, this might be a really basic question. Can you hear me?
Now I can hear you.
So does autophagy increase with age or decrease with age?
Yeah. So my
Yes.
reporter increases with age. No other published results say that that would be consistent. Everybody else shows it decreases with age, because it makes sense, right? If chaos is increasing, if stuff's aggregating and breaking, that would probably be the result of an inefficient cleanup mechanism, right? If autophagy breaks, that would be a logical connection to proteins, increase in dysfunctional proteins. So the thing I'm trying to figure out right now, and this is where my observations are simple on the surface and could be much more interesting once I figure out how to study it more effectively. The accumulation I see is the result either of an increase in autophagy or breakdown in degradation. So just as counting dots measures accumulation of vesicles, and that could either be flux through the system or a blockage in the system, my assay also could be reporting a blockage in the system. It's just at a later step. So it still measures flux in a different way, but if you break the last step, if you can't degrade the protein, it'll accumulate and look like there's increased forward movement, but really there's decreased movement out of the system. So I think it's not broken. So I can deacidify vacuoles. I can force a break in the system, and I see that causes a change. So breaking it makes it worse in old animals. So it's not that it's broken, it just may be that it's not working quite as well.
But we having a hard time breaking down the jellyfish proteins?
Right. And so those GFPs might stick around for that reason. But I can't say whether it's not a combination of the two.
Right.
And that's where the science and the flux and the balance becomes the tricky part of it all. Yeah.
Are your variables the same with other research? Or is there anything else? Or you just said that?
Yeah. Yeah. Each model organism has its own set of variables that throw you off, right? So the worm, so a variable in this system would be, the basic biology of cells is the same, yes. So the way the worm cells work and the way your cells work and the way the mouse cells work, roughly equivalent. In fact, one of the reviewers said, this same reporter has been used in mice and in other systems, and why is yours different? And in a way it's not different because I'm banking on the fact that those two cell types function similarly enough that I can say it works here, it should work just fine here. So I didn't do as much validation as the mouse people had to do because I was using a tool they had already proved. But worms don't require us to deal with IACUC, so you don't have to convince people you aren't harming them, but they do have their own set of trickiness. So they've got this cuticle, which lets them live in the soil. They live in rotting fruit, basically. Most of the model organisms that we use in science come from compost, right? Worms, flies, Drosophila, fruit flies, yeast grows on rotting fruit. I don't know what it is about rotting fruit that's so hospitable. Mice, I don't know, mice might be an outlier. So they've got this thick cuticle, it makes it hard to get drugs in. So I can treat them with this drug that breaks the final step, but I don't know whether the amount I put in the liquid is the amount that their cells feel, because they've got this cuticle protecting them from drugs. So it makes it really hard to get in. So the variable that I have to deal with, that people dealing with cell culture don't have to deal with, is that I don't know what conditions my cells are feeling. But I can starve them and put them in anoxia.
There's no way of measuring?
No. Well, okay, so you can measure a response. So I could take a dose curve and do a dose and time course and say the responses I see in worms are similar to, this point looks like this point in mammalian cells. So I could take a culture system that didn't have a cuticle, look at the responses there, and then sort of map those over to the worms. But that just sounds like a lot of work to me. But that would be the way to do it. Yeah.
Please, thanks.
So aging is this process of increasing entropy or kind of continuous decline. Do you think it's possible to have an infinite lifespan here?
Infinite. I have.
Or what is the maximal possible lifespan you think? And what do you think?
Yeah. A lot of people with a lot of money who want who want answers to that. I don't know. I mean,
Is it open-ended?
I want to say no, and I'm trying to figure out whether that's me saying that as a human or me saying that as a researcher. Given how many things go wrong, I don't know if we could figure out how to fix enough of them well enough to live much beyond 130. I mean, the thing is, we've got most people lived 120-ish, right? So can we bump the average up? Because we don't want to just extend the maximal. We want to sort of float all the ships up on the tide. And we can do better than we are currently with lifespan modulation, but I don't know how much we can boost up that high end. But you will find people who disagree heartily and will swear that we can live to 200.
Our cells individually are probably potentially more cancer cell lines, for example, we cultured for 50 years.
Right.
So I don't believe it either, but the possibility the organism could be perpetuated that long ago.
Yeah. So the idea is that there are cells in culture that we took from cancers, decades ago, and those are just cruising along fine, replicating many more times. So yes, it's good for cells in culture, and I have trouble figuring out what the challenge is when we get to a multicellular organism, right? What is it about being in a body that makes it harder to live forever? I mean, you have to maintain order, and you can't just sort of have, because cancer cells lose a lot of functionality and specificity and differentiation when they decide to live forever.
Cells in culture have the artificially controlled environment.
Right, and cells
everywhere else in the world have complex environmental factors that
That seems to be possible in organisms that control everything. I don't believe it. Can I ask a question another way? Why do you, given that most of us would read an evolution cocktail would reproduce by the time we're 30, why do you think we live so much longer?
Oh, so
Why
this
do you
is,
think, why do you think we're capable of living to 130 years? From a genetic point of view, we know what we need to do by the time we're 30.
yeah, so this is an interesting question that gets at the intersection between aging and evolutionary selective pressure. The idea is selection only happens on things that affect reproduction, right? So if you have a child by the time you're 30, and this is one of the ways people explain aging, things that make you a healthy 30-year-old may not necessarily make you a healthy 50 or 90-year-old. But there is no disadvantage to getting old because your kids are already grown and your genes have passed on. So there is essentially no selective pressure for living a long life. Genetically speaking, there's no benefit to living past your reproductive years. And that's one of the other theories I didn't put up there was this selective switch, the benefit switch, right? The idea that things that work really well when you're young and help you live fast and go hard and reproduce lots end up running you out of energy or whatever, right? Metaphorically speaking. By the time you're old, and so you die less healthy than were you to be not as reproductively fit earlier and not have those sacrifices kick in later. So that's for sure an issue. And so I think this living forever thing sort of requires a reprogramming of more to it than just a gene or two or an organelle or two, because there's an entire genetic program that's been going on for millennia, right?
So you've got three stresses, lack of oxygen, lack of food, and just time. and each of them gave you different results. Can you explain why one would give you result A, B, or C?
Well, I expected, so I use them as controls. People have published on anoxia and on starvation and shown that autophagy goes up. So I expected those to both behave the same. So it was a surprise to me that they didn't. It makes sense for starvation to increase autophagy, because if you aren't bringing in any nutrients, one way to keep producing essential proteins is to break down the ones you already have. Sort of, yeah, self-sacrifice. And then anoxia, it was shown that autophagy goes up as well. I think it has to do with the severity of the stress. I designed both the experiments so that they kill approximately the same number of worms, because that's the way I have to measure how stressful it really is. So it was roughly similarly deadly, which is the only way I have to say it's the same. And the fact that the intestines respond differently tell me, and this is matches what's in the literature, that the intestines are a site of organismal effect. So metabolism that changes in the worm intestine has effects in the whole critter. And we're still, that's fairly new research, and they're still trying to understand that. So it tells me there's tissue-specific differences, and I don't understand exactly why.
Does it matter if you die from have these problems start to replicate if it's a problem in your skin or a problem in your intestines? Which one do the worms fight off best? I don't know exactly how to ask that question. So one affected their intestines and one affected their skin. And is one better or worse for the worms or us?
Oh, like pick your stress?
Yeah.
You have to die by one stress.
I don't think we can do anything about the clock, but I think we can do something about the food and the oxidation. So.
Yeah. I think that time will tell. I don't think we know for sure yet. I mean, the question is, what do you decide is most important? Do you want to keep your neurons or keep your muscles? And I would vote for keeping my neurons. So then the question is or would I? Maybe that's debatable also. Which stress affects those? And that's the kind of tissue-specific response question that I don't think we know yet. Because we're still trying to figure out if you stress a mouse, what happens as far as tissue-specific responses. And I don't think we can say. Which doesn't really answer your question at all, but.
Okay.
Yeah.
I don't know if in worms you have such a thing as a strain.
A strain? Yeah. Yep.
So are there strains of worms that are different in aging?
Right. So are there strain differences in aging? I'm trying to remember the folks who do strain stuff. Yes. We don't think about it quite as much in worms as people have been thinking about it in mice.
Right.
So there have been some really good studies showing if you do, for example, caloric restriction in mice, it has huge positive effects in 24 strains and moderate negative effects in 12 strains. And what's the difference there? Many times, worms are hermaphrodites. Cool fact. So most worms have both male and female reproductive organs. There can be males generated in times of stress. So we don't have to worry about sexual dimorphism, but other studies do have to separate it out by males and females, and often there's a sex-specific lifespan effect of any particular treatment. So that's a significant concern, and we tend to ignore those because we have hermaphrodites and all use the same strain.
That's pretty wild.
Yeah.
This is pretty specific data of your study. I'm sure you're familiar with Cynthia Kenyon.
Yes.
In her study on worms about aging. So they came to a conclusion that green tea actually expands.
That what?
Like green tea.
Green tea.
In humans. Now my question actually is not about that. It's about what you said when you were presenting the different types of animals and their lifespan related to their weight, heartbeats, energy consumption. We all know that energy working out degrades, apparently, but prolongs your life in the long term. So do you think there's a balance to strike between energy consumption or energy efficiency and the effectiveness of our organisms during our lifespan? Because you were talking about health span and lifespan,
Right.
two different things.
Right.
That's what I want to ask about.
So the effect of exercise on metabolism and lifespan.
So degrading cells
Right.
is actually somewhat beneficial for a longer or healthier lifespan.
Right. So there's a paradox there, right?
It's a little bit of a contradiction,
Because you-
which is obvious, but I want to talk about that a little bit.
Yeah. So the paradox there is exercise, and when you exercise, you essentially rip apart muscle cells, right, and ask them to be put back together. And you use energy. I mean, you're using energy when you exercise, so you're in theory generating more.
And your heartbeat goes up.
And your heartbeat goes up, right? So I think that's partly a difference between acute and chronic conditions in a way. So your heartbeat goes up a little bit, but then it goes back down to resting levels, and it strengthens itself, so then your heartbeat over time slows, right? So a transient increase in heart rate results in an overall decrease in heart rate. And so because it's like a muscle, right? You've made it stronger by exercising it. But that effect doesn't have as much to do, I mean, those graphs with the heartbeat rate were actually underlying a whole set of metabolism effects. Right? Heart rate is the result of all these other genetic preconditions that set it up to live fast or live slow. So you can't do much to change your place on that graph. What you can do is exercise, and there's a bunch of good things that does for your aging. And there are many people who think that's really all you can do to slow your aging is exercise. And that's the best way to do it. Incidences of cancer and disease all go down maximally at about 30 minutes of exercise a day. After that, you get a little bit more benefit, but it's not super much. But you pretty much reduce your risk for everything if you exercise 30 minutes a day. And so that's one of the reasons it increases your lifespan, because you decrease your propensity to get sick other ways. It doesn't change your DNA necessarily, but it changes how your body responds to living.
It's a basic question, but like when they're, people have different, maybe I've heard people have different levels of metabolism between individuals, and then sometimes we'll also make the remark like, gosh, that person has always a lot of energy, that person has maybe less energy, but is there any truth? Can you even spot somebody who has a high or low metabolism, or are those completely different concepts that you follow?
So the metabolism that I've been talking about versus the colloquial use of the
Kind of energetic
right,
person
energetic.
who's always on the go.
Yeah. I don't think so much. I mean, yes, you can, right? Because people who are energetic probably change their basal activity level, and so use more energy for sure. I don't know studies looking at whether the baseline begins the same for everybody, and then it doesn't begin the same for everybody, and I don't know how much you can affect its divergence.
And the telltale of metabolism is simply people's, what
It's-
are kind of the vital signs to assess someone's metabolism?
So to check a metabolism, you put an animal, I don't think we do it to humans very often, in a cage in which they measure their energetic output. So you measure everything that goes in and everything that comes out of an animal. You measure the heat it puts off, you measure its waste, you measure how much it breathes. I mean, you can measure the entire thing. So then you calculate kilojoules or kilocalories or whatever per unit of body mass per time, and that's metabolism essentially.
So if you're does that mean that if you're a cold, I'm sorry, this is my last question. If you're like a cold sleeper who is throwing off heat like crazy, then you have a higher metabolism than kind of somebody who's always needs to come
cover up, or is it?
Colloquially, yes, but I don't think it actually is as explanatory as we want. Because there's a bunch else that affects how you perceive your own internal temperature.
Oh.
So that gets into the brain, and that's a whole nother talk.
Wow. Okay.
Well, it's been about an hour. Are there any burning last questions before?
I want to talk a little bit longer. Do you have any kind of useful definitions of difference between health span and lifespan? You kind of touched on it and then shifted a different direction.
Health span is how long you live without getting really sick, and lifespan is how long you live, period. And beyond that, we're sort of working to figure out how to operationally define health span and why to define it. And this gets back to the funding and the regulatory questions. Like, every study that looks at health span defines it in their own way for their own endpoints. Biomarkers is a big challenge. Are there biomarkers of aging? Are there things I can draw from your blood or your urine that tell me how old you are? And can I, from that, determine your health or your lifespan? And you can't yet. But that would be, so different studies. One study will look at how long you can, getting out of a chair without using your hands is one of the best predictors of how old you're going to live, right? Is that health? Is by the time you can't do that anymore, does that declare you unhealthy? Is it cancer? Is it heart disease, diagnosed heart disease? What about undiagnosed heart disease? So that gets trickier, but simple.
Any last questions? One.
So all the genetic code is written in the DNA.
Yep.
So as we all know, we start from a cell or two cells interacting and all that, and we start multiplying, dividing, whatever, becoming an organism. Is there any hint that the DNA might contain data about one's lifespan?
So the question is, we are all DNA. We all come from the genetic code, right? So it's a series of 4 letters that spell out you, which still kind of blows my mind. Can you read that code and decide how long you're going to live, essentially?
Is there any hint?
Are there hints in there? I mean, yes, there are hints in there. Do we know how to read them and interpret them? No. So we're given this book with this, pages and chapters and this tome of information. We don't understand how to read it because you do these cool experiments where you'll sequence the entire genome of centenarians. There's a bunch of people who focus their work on centenarians. I've got to tell you this because it's cool, and then we'll go. Centenarians, why do they live to be 100? In fact, one of the guys who studies it because all of his family died at age 60 and all of his wife's family lived to be 100, and he's like, damn it, I'm going to figure out why that is. Turns out you can sequence their DNA, and they have mutations that would be associated with cancer, but they don't have cancer. So their DNA is not cleaner than ours. Their DNA is still full of errors. It's not that they're better at keeping their DNA clean and error-free. It's just that they aren't affected by those mutations. And why that is going to be fascinating to understand. So no, there's a bunch of things we do know about the genetic code, and there are some genes that you could look at and say, whoop, that's not a good indicator. But there's a bunch of other stuff that doesn't tell us.
I don't know that. But do you think, what do you think?
Oh, what do I think?
Is there any data? Is it possible that the DNA would contain any kind of data, any kind of hint about one's lifespan?
It is very possible that the DNA contains hints about lifespan, yes. But you ask the guy who loves mitochondria, and he'll say, no, it's all in the mitochondria, so I don't know.
Awesome. Okay, thank you very much. Let's thank you.
Great, thank you, guys.